Hello class, today we're going to be talking about DNA. And the first thing that we should be able to discuss is going to be able to summarize the relationship between genes and DNA. Then we should be able to describe the overall structure of DNA molecule. First off, let's start with molecular genetics. What is molecular genetics? Okay, molecular genetics is actually the study of chemistry of the material of inheritance. They study the structure of DNA, and they look at how the DNA contains information to construct the molecules needed for life, which would be proteins. We're going to look at a couple different experiments here that have to deal with DNA. First, we're going to start off with Griffith and transformation. And what Frederick did was that he started researching pneumonia. And what he did with this experiment is he actually had a disease-causing bacteria. He injected that into a mouse, and it killed the mouse. Then he had harmless bacteria. He injected that into the mouse, and the mouse stayed alive. Then he heat killed the bacteria, so he killed the disease-causing bacteria, and he put that into the mouse, and the mouse stayed alive. After that, he plated that as well, and there was no growth, so that he knew that the bacteria was dead. He then added the heat-killed bacteria to the harmless bacteria, injected that into the mouse, and then it killed the mouse as well. He then plated the bacteria from that, and there was growth again. So then he concluded that transformation was there. And so, like I said before, he mixed the heat-killed disease-causing bacteria with the harmless bacteria, and the mouse died. So a factor that formed that the heat-killed bacteria had transformed or changed the harmless bacteria into disease-causing ones. Transforming factor may be a gene. After that, Ivory looked at DNA. So Oswald Ivory was in 1944, and he repeats Griffith's experiment. But he looked at what part of the heat-killed bacteria was responsible for the transformation, and he came up with that the DNA stores and transmits the genetic information from one generation to the next. Hershey and Chase's experiment. What they looked at in 1952 is they studied viruses, which here is a virus. We have the tail, and then we have the end tail fibers, which are all proteins, and those are going to attach to the cell wall. And viruses are a little bit different from bacteria. Bacteria will be able to be killed off from antibiotics, and viruses cannot. So viruses are non-living particles smaller than a cell, and they can infect living organisms. And what these two looked at is actually a bacteriophage, which is a virus that kills bacteria. So it eats bacteria. And it's composed of the DNA, RNA core, and then a protein coat. So we have the DNA and the RNA up in the head, or the core here. We have the tail, and the core actually goes into the cell wall, and then that way it injects its DNA and RNA into the cell, and that's what kills the bacteria. So they looked at two different bacteria. So they have the same bacteria here, and what they did is they took two different phages, one with phosphorus in the core and one with sulfur on the outside. And they looked at if the bacteria would be infected by the phage. So here we have the phages infect both the bacteria. You can see the orange dots, that's the sulfur, and the pinkish purple dots, that is the phosphorus, and how it goes into the bacteria and injects its DNA into the bacteria. The sulfur is still on the outside, so it was not injected. However, the phosphorus was injected. So radioactivity inside the bacterium. And then this one had no radioactivity inside the bacterium. Hershey and Chase concluded that genetic material in a bacteriophage was DNA and was not a protein. 3 critical things that genes do. They carry information from one generation to the next. They carry information to create proteins. and they must be easy to copy. Now let's look at the structure of DNA. DNA is a molecule made up of nucleotides, and there's thousands of repeated smaller parts. Nucleotides are made up of 3 parts. We have a 5-carbon sugar, we have a phosphate group, and we have a nitrogen base. And we already talked about all of this in chapter 2. So here is our phosphate group. We have our 5-carbon sugar here, and then we have our nitrogen base. So some components in the structure of DNA. Okay, we have 4 nitrogen bases that make up the rungs of DNA. So we know that DNA is a double helix, and it kind of looks like a ladder. And we have the rungs in the middle, which are the connections between the nitrogen bases. We have adenine, which is the A, guanine, which is the G, cytosine, which is the C, and thymine, which is the T. A and T are always going to bind together, and C and G are always going to bind together. So Erwin Kofgard, American's biochemist. In any sample of DNA, there's percentages of cytosine and guanine are always almost equal, and the percentages of thymine and adenine are almost equal. So that just shows that they do bind together. The bases are bound in the middle of the the bases are bound at the middle of the DNA molecule in complementary pairs. Each nitrogen base will only bond with another specific base, which is called base pairing. Like I said already, adenine bonds with thymine, and cytosine bonds with guanine. These bases are connected at the middle of the DNA molecule by a weakened hydrogen bond. So we all know what hydrogen bonding is. It's when we have one hydrogen, okay, that is attracted to a oxygen, nitrogen, or phosphate of another one. X-ray evidence. In early 1950s, Rosalind Franklin used X-ray diffraction to view the structure of DNA. It's an X-shaped pattern, suggests 2 strands in the structure. Crick and Watson made a model of DNA, which was a double helix, so it had 2 strands that are wound together, and it looks like the picture over here, which is also a twisted ladder, or also known as a spiral staircase. So that is the structure of DNA. You need to know that it is a double helix. You need to know the nitrogen bases, and that's pretty much it. Just kind of make sure you know what the experiments were for the discovery of genes, and that should be everything that's going to be on this section of the test. Have a great day.